set limits to our imagination. Good afternoon, everyone. I hope you are all doing fine in such unprecedented times. The world is full of gems and diamonds, and we have the pleasure of having some of them here today with us. I, Siddhant Sharma, on behalf of Biochemistry Department, Deshbandhu College, University of Delhi, feel honored to welcome our esteemed speaker, our respected principal sir, all the faculty members, and my fellow students on this auspicious occasion of our departmental fest, Biospark 2022. It's my pleasure to walk you through the conference activities and introduce you to today's speaker, Dr. Mahavir Singh, an associate professor at IISc Bengaluru, that have graced us to share their knowledge and enhance our mental horizons to their wonderful talk. Biospark has been a great platform for our students to share our innovative ideas. It has thus inspired each student to simulate their exploratory regions of their brain and to come up with exciting ideas and question the fact that whether science is an organized knowledge or not. We feel grateful to celebrate this event year after year, and by showing our full enthusiasm and joy of learning, I really do hope that our students, make the best out of it. Due to the changing times caused by COVID-19, the major crux of this event and others is the use of digital platforms like Zoom to conduct and organize various events in our department. We, the biochemistry family, were successful in orchestrating the events for last year's 2021 Biospark, which included InnoSpark and Cell Diagloton, where more than 20 entries were received in each category. Students had the opportunity to showcase their creative side by drawing a cartoon depicting a biological phenomena, which gave an InnoSpark, the other event, gave them the opportunity to present their effective ideas through the medium of presentation. The event's success reverberated through the overwhelming response from the students. And today in Biospark 2022, we are organizing Sci-Idea, which is to design the cover page of Biospark 2002 magazine. The event Sci-Idea provides a platform to all our creative tinkerers to design and depict the role of science and technology in advancement of our country through the medium of art. Thus, Biospark activities have been a great source for students to express their ideas in an innovative yet time-bound manner. So, no auspicious occasion starts before praying gratitude to God, and hence I would like to begin this event with the blessings of Goddess Ma Saraswati, the goddess of knowledge.
श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा। शुक्लां ब्रह्मविचारसारपरमाम् आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीम् अभयदां जाड्यान्धकारापहाम्। हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थितां वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदाम्।
The mic seems to be muted.
Sorry. Moving along with God's blessing, I would like to call our teacher in charge, Dr. Kamna Singh ma'am, to set off the proceedings for the today.
Thank you, Siddhant. Am I audible?
Yes, ma'am.
Am I audible?
Yes.
Very, very warm welcome to everyone. Friends, today I'm feeling doubly honored, blessed and proud as guest speaker, Dr. Mahavir Singh, and my partner convener, Dr. Meera Kaushal, have been my own students, reminding me my early young days in this college. Since then, it has been a marvelous journey of mine till now. It's my utmost pleasure to welcome our guest speaker, Dr. Mahavir Singh, Associate Professor, Molecular Biophysics Unit, Indian Institute of Sciences, Bangalore, our Principal, Professor Rajiv Agarwal, Vice Principal, Professor Kamal Kumar Gupta, IQAC Coordinator, Dr. Aditya Saxena, DBT Star Scheme Coordinator, Dr. Indrakant Singh, Dr. Meera Kaushal, Founder and Financer for Biospark magazine, Dr. Meera Kaushal, Convener, Dr. Prabha Arya, Dr. Vanshika Lum, co-conveners, Dr. Rajni Jain, Dr. Preeti Karwal, Dr. Astha Giri, Dr. Garima Singh, Members Organizing Committee, Dr. Manisha Sanger, and Dr. Aparna Notyal, judges for Sci Idea competition, esteemed colleagues. Last but not the least, my sweetheart, dear students, for today's webinar on structure and assembly of toxin, antitoxin systems, intracellular molecular time bombs in bacteria, which will be followed by inter-college Sci Idea competition for cover page designing of Biospark 2022, magazine with the theme, Role of Science and Technology for Atmanirbhar Bharat. My huge admiration, appreciation, and of course, all-time respect to our principal, sir, Vice Principal, sir, Dr. Aditya Saxena and Dr. Indrakant Singh, to take our college to new glory and newer heights and giving new status and image to our college. Friends, our principal, sir, is always a great source of motivation, inspiration, and encouragement to all of us. Sir's contribution in the academic growth and infrastructural progress is a notable chapter in the history of Deshbandhu College. Now coming to Dr. Mahavir, who had been a star student of this college, passed out from the Department of Biochemistry in the year 1999. For a teacher, no moment can be better than seeing the progress and listening to a speaker who has been your own student, feeling really elevated to have you in your parent institution, which gave you wings to fly high and wisdom to shape your future. Best wishes, Dr. Mahavir, for your upcoming endeavors. If it would have been this would have been really great if you could visit our college now with a newly established I4 center under the DBT Star scheme, where all the latest modern equipments are kept, a newly constructed department with modern lab and teaching rooms with LCD projectors, etc. My sincere thanks to Dr. Mahavir to spare your valuable time and being with us. My heartfelt thanks to Dr. Varsa Vaveja, social media champion, for publicizing the event widely for maximum participation. Thanks to dear judges, Dr. Manisha Sanger and Dr. Aparna Notyal, for taking out valuable time to judge the Sci Idea competition. Thanks, Bonika, from the bottom of my heart. As always, you are so dear to me. Thanks to all my departmental colleagues for being with me always and providing me strong support system. Thanks to my dear students, Siddhant and Kanishk, for taking care of anchoring part. Huge thanks to Ayush for making the poster so artistically. Thanks to all the participants of the Sci Idea competition and congratulations to all the winners. Last but not the least, my huge heartfelt thanks to all my worthy colleagues and dear students to be the audience of the webinar, as without audience, any event is fruitless. I wish a great success to this event. Thank you, everyone. Stay safe and healthy. Over to you, Siddhant.
Thank you, Dr. Kamna ma'am, for providing us with a great start to this event. Now, I would like to invite our Honorable Principal, sir, to speak a few inspiring words.
Thank you, Siddhant. Thank you, Dr. Kamna, for the kind words about me. Dear colleagues and dear students from our college and other colleges, I welcome you all on behalf of entire Deshbandhu College fraternity on yet another edition of Biospark, which is organized by Department of Biochemistry of our college. On this occasion, I would like to extend my greetings and welcome to our distinguished speaker of the day, Dr. Mahavir Singh, Associate Professor, Indian Institute of Science, Bangalore. As Dr. Kamna said, it's a moment of pride for us, as Professor Mahavir Singh is an alumnus of Deshbandhu College. We are very happy to have you amongst us. Professor Singh today is going to deliver his talk on toxin, antitoxin system and its implication in certain specialized area. Dear students and friends, Biospark, as has been told, is the Inter-College Academic Festival of the Department of Biochemistry of Deshbandhu College, wherein the scientific aptitude of students is celebrated through means of creative channel. It gives an opportunity to the students to interact with eminent researchers from the field of biological sciences and increase their interest as well as knowledge towards the subject. The department has been organizing Biospark every year in form of seminars, symposia by national and international speakers. It also hosts various student activities like innovative ideas, presentations, cell dialogue tools, poster presentations, debates, panel discussions, to name a few. This time, there is also an Inter-College Biospark magazine cover page designing competition, Sci Idea, on the theme role of science and technology for an Atmanirbhar Bharat. Biospark is an attempt to bridge the gap between undergraduate studies and research studies and motivate the students towards higher studies in the field of life sciences. In my understanding, as far as today's topic is concerned, toxin antitoxin system are being understood as a key bacterial defense mechanism against invading viruses, antibiotics, and other environmental stress. Well, I will not go further than this and hope Professor Singh will carry forward and enlighten us with the findings and observations in this area. Before I end or conclude, I would like to thank our Vice Principal, Professor Kamal Gupta, conveners of the program, our TIC, Dr. Kamda Singh, Dr. Vinita, DPT Coordinator, Dr. Indrakant Singh, our IQAC Coordinator, Dr. Aditya Saxena, co-conveners of the program, Dr. Prabha, Dr. Vanshika, Dr. Rajni Jain, Dr. Meera Kaushal, and all the students as well as faculty members of the organizing committee. I wish you all success. Thank you. Over to you, Siddhant, now.
Principal, sir, for sharing your pieces of wisdom to all of us present here. Now, I would like to invite our respected Vice Principal, sir, to address the audience.
Siddhant, has he joined? Vice Principal sir has joined or not?
No, ma'am.
I don't think so, ma'am. Sir had joined, Vinita?
I think he has not joined. Probably he must be forgetting. So
you can go ahead then. You can
yes.
Go ahead.
Please.
Okay, ma'am. So without any further delay, I would like to call Dr. Meera Kaushal, ma'am, to introduce our guest speaker and begin the most awaited event for the day.
A very good afternoon to all of you. I, Dr. Meera Kaushal, on behalf of Biochemistry Department, welcome you all to the first webinar of the year 2022 for our Academic Fest Biospark. It's a pleasure and privilege to introduce our guest speaker, Dr. Mahavir Singh, an Associate Professor of Molecular Biophysics Unit from Indian Institute of Science, Bangalore, who is also an alumnus of a prestigious institution. He will be sharing his insights on the structure and assembly of toxin antitoxin systems, intracellular molecular time bombs in bacteria. Dr. Singh is a scientific luminary who has made significant contributions to the field of molecular biophysics. Dr. Singh completed his bachelor's degree in biochemistry from Deshbandhu College in 1999, followed by master's from University of Delhi, South Campus, and PhD in biochemistry from Max Planck Institute, Germany. He did his postdoctorate from UCLA, USA, under his Professor Julie Pagon. He was bestowed with numerous awards and accolades, like Crystal Award from Chemistry Nobel Laureate Professor Dr. Robert Huber, and Research Excellence Award from MDI and Biochemistry Department, UCLA, Chancellor Nomination Award for Postdoctoral Research Excellence from UCLA, and Ramalinga Swami Re-Entry Fellowship from DPT, India, during his academic career. Recently, this year too, in November, he was awarded with the prestigious Sir BSD Star Award for Excellence in Research adding another feather in his cap. Congratulations, sir. He has currently 9 PhD students and 2 MSc students under his supervision, and he is in the charge of NMR facility of ISC Bangalore along with the molecular biophysics unit. He has published many research papers in peer-reviewed journals with high impact factor, such as Journal of Biological Chemistry, Nucleic Acid Research, PEPS Letter, and many more. Now, I would like to invite Dr. Mahali Singh to give his presentation without taking any more time. Over to you, sir.
All right. Thank you, Vinita. So I will share my slide. Am I audible?
Yes.
Yes,
Okay.
yes.
Okay. Just 1 second. Okay.
See, sir. You can see, sir.
All right. So my slides are visible, right?
Yes, sir.
Okay. Okay. Thank you. So thank you, Vinita, for this rather very elaborate, introduction, which I'm not used to, but thank you very much. Also, I would like to thank, Principal Sir for the Professor Agarwal for kind invitation, to give this lecture, and of course Vinita and Kamna ma'am for coordinating this. I'm thrilled. I'm very happy to be here and talk about some of the work that we are doing here at IISc, and I'm going to share that with you. I just don't know how to where to start today. A lot of memories of our college are flashing in my mind since the time I realized that I have to give this lecture. All those memories, fond memories, are coming to my mind. So let's start. Okay, just 1 second. Yeah. All right. So first of all, let me introduce further, my time at Deshbandhu College. Okay. So as Vinita has mentioned, I was there for my BSc Honors Biochemistry, our prestigious course at Delhi University, from 1996 to 1999. Okay. It used to be very, very difficult to get into this course, right? So you had to score very high in your 12th board exam, and then then only you could make it. And at that time, the intake used to be very small. Sorry, I'm going to take some time to in this slide. Then I'll come to the research. Okay, so anyway, so I was lucky enough that I was admitted to this course, right? And we had 4 wonderful teachers. Their teaching literally is staying with us even now. After passing out from Deshbandhu College, I went to South Campus for master in biochemistry, and there our professor Professor Devi Sarkar used to tell us that you have learned everything in your BSc course at Deshbandhu College, and it is true. After that it was just we would study a little bit in more detail and with more interest, but the basics were set there at our college. Okay, and with these 4 wonderful teachers at that time, I know now we have new teachers, Meera ma'am, Shashi ma'am, Rajni ma'am, Kamna ma'am. We really our basics were really set in our college. And I always, wherever I've been so far, I always say that, this was such a wonderful course, very wonderful curriculum, even at that time, very modern. I'm sure it has gotten better now. So it was we had a lot of fun, and we learned a lot. So, my batch, this is, these were my batchmates, Palani, Pooja, Richa, Sachin, Sapna, Shweta, Tina, and then me. We had wonderful time there, and I know this lecture, and you are celebrating Biospark 2022, but let me remind all of you that our batch in the second year in '98, we actually organized the first Biospark 90. We still remember it as Biospark 98. So in '98, our batch started Biospark, and a lot of credit, should go to Sachin. This guy here, Sachin Malhotra, who was very, enthusiastic for doing a fest, and he galvanized, and we said, okay, we'll do this fest, right? And yeah, we had a lot of fun, and I'm so glad, that this Biospark is continuing and getting even better, right? So it's wonderful. It's wonderful. So yeah, okay. So with this, I mean, I'm sure I would love to visit college physically, and come and see all my teachers, and now new colleagues once it is possible, right? And yeah, I am from Delhi, so I keep coming there. So definitely next time, if I'm whenever I'm there and college is open, I would love to come. Okay. So now with this, I will now talk about give a lecture on today's topic, and yeah, the topic, the title that I made is Structure and Assembly of Toxin Antitoxin Systems, also known as TA system for short, which are considered as intracellular molecular time bombs in bacteria, and I will explain what I mean by that. By the way, if it will be difficult for me to see if anybody is raising hand, so I request Vinita that if somebody has raised hand, you can interrupt me, and I will answer the question.
So we have asked the students to write the questions in the chat box. So after your talk, we'll take the questions.
Okay. So only after the talk?
Yes, sir. Yes, sir.
Okay, no problem. Okay, all right. So yeah. So, principals are already mentioned that, what TA systems are like in a very simple way. We all are, going through this pandemic caused by SARS-CoV-2 virus, right? And I mean, viruses can infect human. We are living the pandemic right now. But, bacteria, right? Virus is actually trouble, or viruses infect even bacteria, right? So they infect all forms of life, including bacteria. And as a result, bacteria they have developed elaborate antiviral strategies to protect themselves against these phage attacks. Okay, the bacteriophage attack. In fact, the statistics are quite remarkable that almost every second, a bacteria is bombarded with a phage. Okay, so it's all the time they are under phage attack, so they need to survive, and of course they survive, and so they have bacteria have an elaborate anti viral mechanisms in them. Okay, and some of these mechanisms you would know already, I'm assuming. For example, restriction modification system, right? So you have through which, I mean, you all would have used some restriction enzymes and in your recombinant DNA work and so on, right? So there is a restriction modification system where the bacterial enzymes can cleave specifically viral DNA, but it protects its own genome, right? So invading virus is the DNA is degraded by the system, right? Then you have CRISPR-Cas system, which everybody is knowing these days, right? Where it's an adaptive immune system in bacteria, where not only that the invading virus is eliminated by bacteria, but it also keeps a memory of this virus, so that next time when a viral attack happens, bacteria is better prepared to counter it. So there are numerous other mechanisms which I am not well aware of, but there is also another mechanism called abortive infection, I mean, here, what happens is that when a phage, a bacteriophage attacks bacteria, the certain mechanisms in the bacteria will get activated, and bacteria will undergo what we call as altruistic cell death. It will kill itself, right, before virus or virus could assemble and proliferate further. Okay, so this TA system that I'm going to talk about today actually fall under this. Okay, so initially they were all identified as abortive infection system. Okay, and this is one anti-phage strategy in bacteria. So let me introduce. So by the way, in general, our lab here at IISc, we study protein nucleic acid interactions, protein-protein interactions in several important cellular systems. So today I'm going to just focus on TA systems here. Okay, the toxin antitoxin system. So what are TA systems now? Okay, I already mentioned to you that they fall under this abortive infection, ABI system, abortive infection system, and they are activated during phage attack, and they help during bacteria to survive during that time. Okay, now the TA system, as the name suggests, it consists of a toxin and an antitoxin. Okay, so typically toxin is a protein, and antitoxin is either an RNA or a protein. Okay, and they are part of an operon. Okay, so you all would also know what an operon is. Basically, you have two genes, antitoxin and toxin, under a same one promoter. Okay, so this is an operon. And let's say that if bacteria somehow makes toxin, free toxin, it is toxic for bacteria. It inhibits vital cellular processes in bacteria itself. Okay, and it will either kill bacteria or inhibit bacterial growth. Okay, that is why we call it as the cellular time bomb, because it's sort of a bomb that bacteria is keeping within itself. Okay, and at times bacteria actually unleash it or activates it when required. Okay, but on the other hand, you can also imagine that if we at will in some pathogenic bacteria, let's say in tuberculosis, for example, if you can activate these toxins, okay, you can actually kill bacteria, right? So that is the motivation that to study the system, that we can somehow activate them with a small molecule, with a drug, and so on. And so that's why, as they're studying their structure, their assembly is important. So so this is a toxin, but bacteria will grow happily. So how bacteria takes care of its own toxin is by making an antitoxin molecule. Okay, so it is either in protein or RNA, as I mentioned. So from the same operon, an antitoxin will also be made, and bacteria will keep the toxin in complex with antitoxin, and this TA complex, this toxin antitoxin complex, is now not toxic. Okay, because in this complex form, toxin is not toxic for bacteria. Okay, the binding of antitoxin makes it inactive. And so this is how bacteria grows in the normal conditions. Under stress condition, okay, so under certain condition, as I told you, actually these systems get activated, meaning the toxin somehow gets freed, okay, from bound antitoxin. And this stress could be several types of stresses. One of that, for example, is our viral attack. So when a phage attacks bacteria, that is a stress. Something happens in the bacteria that will lead to the selective degradation of antitoxin, which will free toxin, and bacteria is going to inhibit its growth. Okay, I'll explain that in next slides. So apart from its role in the abortive infection, okay. So again, if I can just explain for a student, if there is no TA system in bacteria, for example, a phage will attack bacteria, and it will replicate, it will package, and it will release back which will infect more bacteria, right? And that is how the phage infection spreads in bacteria. But if there is a TA system, several of T systems, what people have found is that when phage, infects bacteria, these TA systems get activated. So remember, the TA system, bacteria will maintain TA complex. When phage attacks, we still don't know what happens, but there are certain chemicals or certain pathways they get activated, which will selectively remove the antitoxin and free the toxin. Okay, and this toxin is going to either kill that particular bacteria before this bacteriophage is assembled, replicated and assembled. Okay, or it will go in a a resting state. Okay, so if let's say this often people have shown that this bacteria which is infected by phage will commit suicide, altruistic cell death. Okay, and as a result, the phage cannot propagate further, right? So one bacteria will die, it will help other in the colony of bacteria to survive. Okay, so that is abortive infection. These TA systems have also been implicated in maintaining plasmids. Okay, so many times these TA systems are kept on plasmid, and they make sure that after replication of bacteria, when from one bacteria you make two bacteria, the one without plasmid will not survive, and the one with the plasmid containing TA system will survive. So that plasmid containing bacteria will survive. This plasmid will propagate, and plasmid may have other essential, important genes for bacteria. So they are involved in post-segregation killing or plasmid maintenance. Okay. Now, apart from this, TA systems, several of TA systems have also been implicated in antibiotic, resistance or persistent cell formation under stress conditions such as antibiotic stress. Okay, so in this condition also, what happens? Let's say you have an antibiotic stress given to bacteria. These systems get activated. Okay, the TA systems will get activated. This will inhibit cellular processes in bacteria. Bacteria will not grow. It will not kill bacteria this time, but it is going to go in a dormant state. Okay, until the antibiotic stress is there. Okay, as soon as this antibiotic stress is relieved, again this dormant cell will become active. Okay, it will again grow, and bacteria will survive this condition. This is called persister cell formation. Okay, so these are I mean, there are other functions attributed to TA system, but these are three well-established functions that are attributed to TA system in general. Now, I mentioned TA systems are of different types. Okay, so they have been classified from type 1 to type 6. A very recent review by a very well-known scientist in this area has now included 2 more systems. So I need to update this slide. There are multiple types. Okay, it depends on how they are classified. Basically, first is what is the mechanism of toxin killing the cell, and the other is what is the mechanism of antitoxin binding or inhibiting the toxin. Okay, so based on that, they are classified into mainly six classes or 6 types. In today's talk, I'm going to focus on these 2, type 2 and type 3 TA systems. Okay, because they are well studied and relatively well studied, and also most prevalent of among all 6 types. Okay, so today I'm going to talk about these 2 systems. I will keep a tap of time. I'm told that I have 1 hour, but yeah, so I'll try to finish on time. So so I'm going to talk about type 2 and type 3 TA systems. Okay, in type 2 system, both toxin and antitoxin are protein. Okay, both are protein. In type 3 TA system, it is actually unique in the sense that the antitoxin here is an RNA. Okay, when I talk about this system later, I'll explain this in more detail. And yeah, so we actually we are a structural biology lab. So what it means is that we understand the structure of biomolecular complexes such as protein-protein complexes or protein-RNA complexes. So recently, we have published these two papers, and I'm going to share results from there today. So first, type 2 TA system, a particular type of TA system that we have studied here is called Higg-BA. Okay, and this is the publication. If any student is interested, can have a look at this. Most of the results are there in the paper. And this work is carried out by my student Pankaj and postdoc Vikrant in the lab. Okay, so as I mentioned, this is a Higg-BA type 2 TA system. Here there is a antitoxin called Higg-B, and there is a toxin called Higg-A. Okay, antitoxin is A, which is Higg-A, and toxin is Higg-B. Okay, and this is a ribosome dependent, and so okay, the toxin is toxic for bacteria because often they are RNases. Okay, so in free form they are going to cleave cellular RNA such as mRNA, tRNA, ribosomal RNA, and so on, right? So if bacteria cleaves its own RNA, it is going to basically either slow its growth or kill itself, right? So Higg-B is also an RNase, but it's a special RNase in the sense that it is a ribosome dependent RNA. So meaning it cleaves RNA only when it is bound to ribosome, and it cleaves translating mRNA. Okay, so it binds to the this A site on the ribosome and cleaves the translating RNA. Now, something about type 2 TA system, I already mentioned. It's a complex of antitoxin protein and a toxin protein. They are part of the same operon. Okay, there are several mechanisms by which antitoxin actually, inhibits the activity of toxin. Okay, of course, one is directly occluding the active site of toxin. The other could be more allosteric or steric occlusion, right? Actually, Higg-BA is one such complex where a binding of antitoxin to toxin does not allow this toxin to bind to ribosome, and that is how toxin is rendered inactive. Under stress conditions such as antibiotic stress or phage attack, for example, there is a preferential degradation of antitoxin, okay, which will free toxin under these stress conditions, and then bacteria will undergo either apoptosis or cell death, or will undergo in a dormant state. I'll come to this also, but the operon is also autoregulated. Okay, so you typically this complex also binds to its own operator DNA. Okay, and does not allow its own transcription. This is a negative feedback, of the operon. Bacteria wants to make sure that it is not unnecessarily making the toxin and antitoxin. So actually, if there is enough of a TA complex in the cell, it will bind to its promoter and act as a repressor, or it will not allow its transcription. Okay, as I mentioned, many of these toxins are endoribonucleases. That is how they are toxic for the bacteria, and they can be ribosome dependent and ribosome independent. In this particular work, we studied a Higg-BA a TA system known as Higg-BA, where Higg-B is toxin and Higg-A is antitoxin. So if this toxin is toxic for bacteria, if you want to now, for our kind of work, what we do, we simply take that protein that we want to make. You would have studied, at least in by third year, you would already know recombinant DNA methods. You would put that gene in a plasmid, and you transform bacteria, right? But I'm saying that Higg-B, that toxin, is very toxic for bacteria. So you'll actually not even get colonies, right? You can't make it. Most of the toxins you can't express in bacteria. Okay, so what do you do? What we did is that we cloned both toxin and antitoxin together. Okay, in a same plasmid. It's in a duet vector. Okay, so you have both toxin and antitoxin in the same plasmid, and both will be made simultaneously, and bacteria will be happy. and you get colonies, right? So this is what my student did, and we can actually express it and purify it and so on, and we can purify. This is a size exclusion chromatography column. You would have studied this also. We can purify this as a complex nicely, and also when you run SDS page, right, you can see both Hig A and Hig B, both toxin and antitoxin purified. Okay, so once we have the purified protein, now we can do our experiment. So I'll not go over all the results, okay, just to keep it simple so that you can follow. We solve the structure of this complex, okay, this Hig BA complex. What we so we get crystals, and through X-ray crystallography method, you can determine the atomic structure of protein or RNA or DNA, macromolecule, right? So this is how the complex looks like. You in this cyan color, you have antitoxin. This is our antitoxin. It is dimerizing, right? So you have two molecules of antitoxin dimerized through this alpha 1 helix here at the top, okay, and you have the toxin Hig B bound to antitoxin. So overall, this is a quaternary structure, okay, where you have two antitoxin bound to two toxin. Okay, this is how Hig BA complex looks like. Okay, now free toxin, which is an ribosome dependent RNase, okay, will be this just one monomer of this toxin, right? It goes and binds to a particular pocket on ribosome. When it forms complex with antitoxin, you can imagine this will increase in size substantially, almost 4 times. Okay, and now in this form, it is not able to occupy the ribosome, that binding site, and hence this complex is inactive. Okay, yeah, and and because the active site of the toxin here is actually exposed, but it is useless because it can be active only when it is bound to ribosome, and in the TA complex form, it is not able to bind to the ribosome. Okay, so individually, if we see antitoxin, this is how it looks like. It has a dimerization motif. At the N terminus, you have then the toxin binding region. This is where the toxin will bind, and you have a DBD, DNA binding domain, at the C terminus. Okay, I mentioned that the TA systems also auto-regulate its own operon. So they bind to its own operon DNA, acts as a repressor, and they bind usually through a DNA binding domain. So this is the DNA binding domain in antitoxin. And toxin looks like this in the free form. Okay, it's a classical RNase T1 fold. Okay, yeah, several RNases will have this fold. So when we solve this structure, you are asked whether you can validate it. So some key residues you mutate, and you find out whether your structure is actually making sense. So for example, in bacteria, if you express only Hig B, as I told you, you don't see bacterial growth. Okay, when we mutated key residues, meaning we have now made it inactive. Okay, bacteria are able to grow. Okay, so this kind of work we do to validate our structure. Okay, now something interesting here. The antitoxin, it's typically what we call an intrinsically disordered protein. Okay, it's a labile protein. Okay, in other antitoxin in TA systems, antitoxin actually has an intrinsically disordered part. Okay, which gets preferentially cleaved under stress. So under stress, the current understanding is that during stress, whether phage or antibiotic stress, certain proteases get upregulated in bacteria, and these proteases will selectively cleave antitoxin molecule. Okay, and why they will be able to cleave antitoxin is because antitoxin has disordered region. But this antitoxin is actually quite structured, right? You can see from the one end to the other end, it is quite folded and structured, right? So, but what we observed is that in solution, this is CD spectroscopy. Again, you would have studied or heard, a little bit about this method. We saw that it is quite well folded. Again, basically agreeing with the crystal structure. Through NMR, though, what we notice is that this is a NMR spectra. This is a typical NMR spectra. I'll explain later also. But from this, what we can conclude is that although it is folded, but it has some dynamic in it, meaning, it is not rigid, it is moving. It seems like it is moving, okay? So, also there was a structure of free form of antitoxin, okay, in blue, and our structure in the bound form with the toxin. And we can see that antitoxin molecule actually undergoes a lot of movement, okay? Now, if I show you this movie, okay. Okay. So this is antitoxin. This is in the free form to bound form. If you see, this is actually bound form, and this goes to free form. Okay. So there is a big change in this DNA binding region of the antitoxin, right? The top part remains almost immobile. The bottom part actually moves a lot. And in complex, if you see here, this is the bound form when the toxin is coming close to it, and then the open, which is free form. Okay, so we, through our work, we discovered this dynamic nature of this antitoxin, right? And we believe that this dynamic nature plays a role when antitoxin has to release toxin during stress condition. Okay, so okay. So, we also wanted to study how, the TA complex auto-regulates its own operon. Again, we wanted to understand it through structural means, our biophysical methods, and through our structural biology approach, right? So I told you that, the TA systems provided negative feedback. So the TA complex, generally antitoxin, has a DNA binding domain through which it binds to its own promoter DNA. So, we looked at the promoter of this system, and there are these palindrome, okay? There are these imperfect palindrome sequences, okay? And, DNA binding DNA binding proteins typically bind to palindrome, for the better affinity, okay? So we thought, okay, we will check whether our Higgs-BA complex actually binds to these palindrome. And sure enough, it binds very nicely in these gel mobility shift assays through also, this is what we call this SEC-MOLS, the size exclusion chromatography linked to the multi-angle light scattering, which will tell you about the molecular weight of your complex. We could see that, protein and RNA, protein and DNA forms a complex, okay? So this is just the free protein TA complex, and this is TA complex in complex with DNA, okay? And then we are in a biophysics department, so we understand things from that perspective. So we wanted to understand how strongly the TA Higgs-BA or Higgs-A complex bind to DNA. Okay, so we do that through several means, but this is one method called it's a thermodynamic method called isothermal titration calorimetry. Okay, so it basically you directly measure heat upon a reaction, right? So when protein and DNA are forming complex, either heat will be released or heat will be absorbed, right? And you measure that. Through that, you can calculate these thermodynamic parameters such as dissociation constant, which tells you how strongly two things are binding. Okay, you can convert that to delta G. You can convert, you directly can measure the enthalpy change and entropy. You can also, from the curve fitting, you can also get, the N value and so on. So so this is what we did. And we found that the Higgs-BA complex or Higgs-A, okay, either antitoxin alone or antitoxin toxin complex binds to the promoter DNA with very high affinity. This is in nanomolar affinity, okay? This is with the 90 nanomolar. And this is just Higgs-A binds with the 36 nanomolar. So approximately, 3 times difference, but both the TA complex and antitoxin alone can interact with DNA with a high affinity. So, that is how it acts as a repressor. Okay. Now we wanted to understand further. Okay, we want to understand. We want to visualize how actually, this Higgs-BA forms, complex with the DNA, and if a protein is binding to DNA, this whole idea how it is specific. Okay, if it is a DNA binding domain, it can go and bind anywhere, right? Any DNA. But these DNA binding proteins like transcription factor, repressor, and so on, they are highly specific for a particular sequence. So the whole motivation is to understand how that specificity is generated, right? For that, this is work in progress. What we could do so far is what I'm showing you here. So as I told you, in antitoxin, there is a DNA binding domain. So that is the domain through which it binds. Okay. Of course, when you compare that the structure of this DNA binding domain with the other DNA binding domain, this is a very classical DNA binding domain. Okay, a classical DNA, a protein fold that is present in many DNA binding proteins. Okay, and through which they bind to DNA. And indeed, it looks very similar to the other known DNA binding domains in other unrelated proteins. So in our kind of approach, what we do is we further simplify the system to understand what we can. So we actually made just the DNA binding domain now, and we wanted to use NMR spectroscopy to get an idea about what residues are binding, how this DNA is able to bind to the this DBD is able to bind to DNA. Okay, so we make just the DNA binding domain. Okay, we just made this this small part of the protein, and this is an NMR spectra. This is called a proton nitrogen 15 HSQC spectra. Okay, just briefly, what this spectra gives you is peaks, right? So there are several peaks here. One peak here represents one NH. Okay, so you have when two residues combine to form a peptide bond, right? To form a peptide bond, you have the NH proton, right? So this is the peptide bond here, okay, CONH, right? And this NH is what you are observing in this particular spectrum. What it means is that for each residue, you are going to get one peak, okay? So, for example, this may be glycine 100, or this will be alanine 102, and so on. Okay, so you get one peak for each residue in this type of NMR spectra. So this you can now imagine that this will act as a you can use this spectra to find out which residues are perturbed when you add a ligand to it. Okay, let's say in our case DNA. Okay, so here so if your protein has, let's say, 100 amino acid, you are going to get that many NH protons here, right? Except for proline, because proline will not have NH, it will remain N. It will become N when it becomes part of the peptide bond, okay, because it has an imino group, right? And so except proline, you are going to see one peak for one residue, right? And you can also, what we call, you can analyze this spectra with the help of, three-dimensional NMR spectra, and you can assign these. So now, for example, I know this is glycine 80, this is glycine 79, this is aspartate 138, and so on. So each peak is now labeled here. And to this spectra now, to this protein now, if I add DNA, okay, I see now the shift in peak. Now, what this tells you is that this peak is perturbed, meaning this peak must be involved in DNA binding. Okay, while some peak, for example, this year or this year are not perturbed, right? So only selected peaks are perturbed here. Okay. Now, if you know the identity of these peaks, these residue, you actually can you have the information now which residues are involved in binding. So this is what we did, and what we call, this is called chemical shift perturbation. Okay, so chemical shifts perturbation, and we identified that this phase of the protein, this alpha 7, loop 2, and alpha 8, helix turn helix. Okay, this becomes a helix turn helix motif, and this is the region of the protein that is binding to the DNA. Now, based on all this, now I'm not going to show all the in between complicated results, but we generated now a model based on this NMR results. Okay, results from NMR. And this is how we think that Higgs-BA complex assembles on DNA. Okay, now this is just a model based on, this is a information-driven model. Okay, based on our NMR result, we know that this will be close to reality. Okay, so right now we are in the process of solving the real structure using advanced methods such as cryo-electron microscopy and X-ray crystallography. Okay, and but right now the based on whatever biophysical assays and NMR spectroscopy, we have this model with us. which tells us how this the DBD in antitoxin binds specifically to this palindrome sequence in DNA, and the residue which were perturbed actually are at the interface of binding. Right? Okay, so yeah, the summary of this part of the work is that we solved a structure of a type 2 TA system called HIC-BA, okay, and which revealed its assembly mechanism. We showed that antitoxin is dynamic in solution, and we showed that HIC-BA or HIC-A binds to the operator DNA with high affinity. That's how it acts as a repressor, which has implications for understanding HIC-BA operand regulation. Okay, now here now I'll switch here and I'll talk about our very recent work on type 3 TA system, which is I feel is more interesting than type 2 TA system, because here antitoxin is an RNA. Okay, so now I'll show you some result on type 3 TA system. Here again you have an antitoxin and you have toxin in the same operon, but here antitoxin is not translated. Okay, it is a non-coding RNA, so it is not translated into a protein like in case of type 2 TA system, but here it rather remains as RNA and it acts as an RNA. So the toxin actually binds to the antitoxin RNA. Okay, here not toxin, not protein. Okay, and assembles in a complex. We solved the structure of first E. coli type 3 TA system. Okay, and it assembles like this. Okay, so I'm going to show this result. This paper was recently published, and Mani and Kaveshri, these two students in the lab. Mani has now graduated. I mean, he finished his PhD. They did this work mainly. Okay, so type 3 TA system, I have already mentioned it a few times. You have the antitoxin, which is RNA. You have toxin, which is protein, and both this operon will be transcribed. Protein part will be translated, this RNA will be translated, the antitoxin will remain as RNA. What happens here is, what is cool here is that toxin here is an RNase, okay, like many type 2 TA system, but it also, this is also an RNase, but it also, processes its own antitoxin. So antitoxin has these repeats in it, okay, so it is, the RNA has some, 30 to 40 nucleotide sequence, which is repeated multiple times, okay. Now, what toxin does, it actually, auto processes its own RNA into a single repeat, and with one repeat, then it will assemble and form a TA complex, which is not toxic for bacteria. Okay, so not only that toxin cleaves the cellular RNA, that is how it will be toxic for bacteria, but it also processes its own antitoxin RNA repeats. In between, there is a terminator here, terminator sequence, right? Which makes sure that, in the cell, most of the time you have excess of antitoxin than toxin. Okay, so remember, any amount of free toxin is going to be toxic for bacteria. So bacteria has several strategies to make sure that you have either TA complex, or you have you always have more of antitoxin than toxin. So yeah, there is a antiterminator sequence in the middle to which makes sure that you have more of antitoxin RNA than toxin protein. So so when we started this work, there were no known E. coli TA system, type 3 TA systems, okay, in the databases. So we, based on simple sequence searches, we looked in different E. coli strains, and we identified that indeed many of the E. coli strains actually have type 3 TA systems. So based on their sequence homology, okay, of the toxin protein, we found that they can be clustered into 5 different clusters. Okay, so we named them from cluster 1 to 5. Okay, so basically the toxin of cluster 1 is similar to one another. Okay, and cluster 2 is similar to each other, and so on. Okay, now further analysis of each of these cluster also led us to make this representation, which represents like the how big the toxin is, how many toxi repeats are there in toxi RNA. Okay, so it seems like each cluster is unique. Okay, so cluster 1 is unique, it has 4.6 toxi repeats, for example. Cluster 2 is unique because it has 5.6 toxi repeat, and so on. Okay, so we classify them like that. Now we choose one of the system, and we wanted to further study this experimentally. Okay, and so this is how it will look like if you have to take the DNA sequence. You look for all the element. You look for the toxin coding region, starting from a ATG start codon and ending with the stop codon. You have the antitoxin repeat in cyan color. You find promoter sequences minus characteristic minus 35 minus 10 signature sequences, and also the anti-terminator, this terminator sequence in between toxin and antitoxin sequences. So it has it has every all these elements, right? So now as I told you about when I was discussing Higgs-BA system, right? The toxin alone, if you try to express in bacteria, you will not get colonies itself. Okay, so you can't do anything. When we, in this case, when we put tox N and tox I, by the way, here just like Higgs-BA, right? Here it is known as tox IN, where tox N is toxin and tox I is the antitoxin. Okay, so when we put the toxin and antitoxin, in the same plasmid, okay, also it did not work somehow for this case. So in this case, we had to optimize our strategy. What we did now is put tox N in one plasmid and tox I in another plasmid. So we had two plasmid, two compatible plasmid, which will have different origin of replications and different copy number, and which will maintain, both the plasmid will be maintained to different copy number. You want to have tox N in the low copy number plasmid and tox I in high copy number plasmid, and of course different antibiotic to maintain them, right? And in the using this strategy, we could actually get colonies and proceed with our work. So first thing is whether it is actually a TA system, right? Because these were brand new. We identified them based on sequence homology, but we are asked whether they actually act as a TA system. So this is so this is basically empty a bacteria growing with empty vector. It grows nicely, but when you express now toxin, okay, you see retardation of bacterial growth. So this is proving to be toxic for bacteria. So this shows that this is a toxin. And in this these spotting assays, right? Now again, when you express only toxin, you see that bacteria are barely growing. When you now rescue this, by expressing antitoxin together with toxin, you can see that bacteria they start growing, right? You can also do it with the CFU counts. So so basically, we could then with two plasmid strategy, we could keeping toxin and antitoxin on separate plasmid, we could purify them, okay, and we could purify toxin protein, and we could purify toxin RNA, okay, and this is a ion exchange chromatography profile, okay. So your protein, your protein is bound to the column, okay. Your lysate is bound to the column, and you are eluting it with the salt gradient, right? So you see that the TA complex comes elutes with the high salt here in the middle, but we also get two peaks here. One at the beginning of the salt gradient, meaning this thing elutes at low salt concentration, and at higher salt concentration also you have another peak. When we checked it on the gel, this is a free protein, free toxin, this is free antitoxin RNA, and this is RNA protein TA complex. Okay, so in one prep, actually we could get all three species. Okay, so we could get free toxin, free antitoxin, and TA complex. This was wonderful for us because we need these three components for our work, right? So then, of course, first thing is we took the complex and we you characterize it further and so on, and yeah. So this is just we basically we say that we devise a strategy where if we put toxin and toxi RNA on separate plasmid, compatible plasmid, you can actually get you can develop this purification protocol where you can get complex, you can get free form of toxin protein and free RNA. Okay. Then the complex we actually crystallize. It crystallizes, and through again X-ray crystallography method, okay, we determine the structure of this complex. Now this is how the structure of this complex looks like. In the asymmetric unit, meaning in the crystals, okay, the smallest unit of crystal, we see that there are two, it's basically a sandwich, like two breads of the sandwich, right? So you have one TA complex here, another TA complex here. But this is just a crystallization artifact. Actual assembly is like this, only one side. Okay, so one side of this complex, right? So you have this heterohexamer. Okay, so basically there are three protein, there are three toxin bound to three antitoxin. Okay, this is one repeat of antitoxin RNA. Remember, I told you this has how many 4.4 repeats or so. This is one repeat. So the toxin had cleaved it and assembled. Now it has assembled with one such repeat, right? So toxin now, is assembled with it, and this is how the assembly looks like, which is, in my opinion, quite beautiful. It is a heterohexamer and a trimeric complex, right? It has a threefold symmetry, So I'll go in little detail about this. So this is, if you see only protein, this is how it looks like. Again, this is a classical RNA fold, okay? And the RNA, RNA is quite beautiful here, right? I mean, this is the RNA from RNA repeat from 5' to 3' end, right? What you can see here is that RNA is not simple single-stranded, right? It actually is folded structure, okay? RNA is folded here, and it folds into a pseudonaut structure, okay? It is a pseudonaut structure, pseudonaut meaning, if you actually pull both the end, 5' and 3' end, you will form, you will not tangle, right? So it's a pseudonaut, but through interactions, it is forming into this pseudonaut structure, which is a structured RNA. Okay. Now you will also remember that RNA actually can fold into complex structure. Again, like group 2 intro, and they are actually RNases, they are enzymes, right? So yeah, RNA has this great potential to form into complex structure, and this antitoxin RNA here also folds into a quite elaborate structure, and this is how it assembles, right? Okay, so now if I see now here, what I have done here, what my student has done, this is a sequence alignment of all the 5 clusters. Okay, now if a representative sequence alignment of all the 5 clusters in E. coli that we had identified. Now, if you align all of them, you find that some of the residues are actually absolutely conserved. They are across different clusters, right? All 5 clusters or many sequences, they are absolutely conserved. So we just wanted to see where do they lie on the structure. So if you map now these red, these residues in red on the structure, they actually all are lying where toxin is binding to antitoxin RNA. Okay, so they are important for their function. Okay, that is why they are conserved. Okay, so and for the antitoxin also, if we now align the antitoxin sequence from all 5 clusters here, okay, we see that the pseudonaut forming part, the pseudonaut forming region is very conserved. Okay, is very conserved. So it seems like the structure, having this structure in this RNA is very important for its proper assembly with the complex. Okay, so the pseudonaut structure is highly conserved. Okay, now if we zoom at the active site, remember now this RNA, of course, this is an antitoxin RNA for this toxin, so it will bind and make the protein inactive. But this had been also a substrate for this RNAs, right? So it from its multiple repeats, it has actually cleaved this and assembled with one of the repeat, right? So we can also think this RNA as its substrate, right? So this RNA is also substrate for this toxin. So yeah, so if you analyze the structure now, you can see the active site here. Okay, you can see how toxin binds, how nicely, it is going to be highly specific. So for example, this A nucleotide here is forming so many hydrogen bond with the protein. Okay, similarly, from this side, this A here, this U here are forming very specific hydrogen bonding network. What it means is that if you replace this A with, let's say, a C or U or G, it is not going to be equivalent. Okay, so it's very specific for a particular sequence. Okay, it's going to be specific for very specific for a particular sequence. Now, also what we found is that at the 3' end, you have this cyclic phosphate. This is 2', 3' cyclization. Okay, so basically we caught this in action. Okay, so when phosphodiester, when protein is cleaving the phosphodiester bond, okay, it is this intermediate is generated. Okay, and it is stuck with that intermediate here. Okay, so this is yeah. And yeah, so from recently, just when we were publishing our paper, there was a paper came where they actually through this degradosome analysis, they actually noted that it should have a GAAU cleavage site. Okay, and indeed our structure beautifully explains that why it should be GAAU. Okay, so if you replace the order or the the nature of these nucleotide here, the either protein will not cleave it or it will not cleave it that better specificity. Yeah, and yeah, indeed it actually cleaves it when we make a substrate. Okay, the toxin actually cleaves it very nicely. So we make a substrate with the cleavage site, and it nicely cleaves RNA into 2 pieces. And again, same here, many of these residues we were asked to validate, their importance. And again, through growth assay, okay, we validated many of these interactions. Okay, and they, the result, these our growth assay results beautifully match with the structure. Okay, so if we see that a residue here is important, the lysine, for example, this lysine 33 to alanine. Okay, this lysine actually is binding to the RNA here, right? So yeah, if you mutate that lysine, the bacteria start growing. Okay, so then one thing is I told you one of the greater motivation of TA system is that eventually people think that you can if you somehow can disrupt this assembly in bacteria, while let's say in pathogenic bacteria, if you can disrupt this assembly of TA system, okay, and you free the toxin, it is going to be toxic for bacteria, and you can have an antibacterial therapy. Okay, so that's why understanding the structure assembly is important. So we wanted to understand assembly of this complex. So we remember when we purify this complex, we get free RNA and free protein. So when we again using isothermal titration calorimetry, when we study this binding, okay, of toxin with the antitoxin, we see this biphagic nature of the complex formation. So you have one event here, okay, one binding event with one binding constant here, okay, or dissociation constant rather, and then you have a second binding event. And we looking at the structure, we believe that the 5'. So what is happening here in the structure? You have one pseudonaut, right? And the protein is binding to the 5' end of the RNA here, and the 3' end of the RNA here. Okay, or if you see in the other way, you have the 5' binding a protein to the 5' part and 3' part of the RNA, right? And these two regions are actually not equivalent. Okay, the number of interactions length are different, and that is reflected in ITC. This is a very powerful method. It is reflected in ITC. You get this biphagic curve. Okay, and you can do curve fitting. You fit 2 binding event to this, right? So you have one with the one affinity, the other one with the other affinity, KD value. And yeah, you further validate this by deleting one end at a time. And this is exactly what Manikandan did here. And when you delete one of the end, you can see that it becomes just single event binding, right? And when you delete both the ends, you see that binding is abolished, right? So just the pseudonaut part is actually not enough for generating the tight complex. Okay, so we are still wondering what is the role of this pseudonaut. Okay, what is the role of this pseudonaut? This pseudonaut seems to be very important for binding for the assembly. Okay, in vivo. So based on all that, what we have proposed is that in vitro, what we observed is that if you put if you take a toxin and antitoxin repeat, it will go through this intermediate binding where one end is bound, and ultimately you will generate a primary complex, which we see in the crystal structure. Inside the cell, what we believe is that as the RNA is getting transcribed, the protein toxin will cleave it, okay, it will again undergo through these intermediate where one end is binding. I mean, from our ITC, we show that the binding to the to the 3' end, okay, when the 3' end is intact, is stronger than to the 5' end, right? So that end will be bound first, and ultimately it will snap and generate this hexameric complex. Okay, so this is what we did so far in this project. We showed that E. coli actually harbors type 3 TA complexes. Okay, these E. colis are of different different types. Okay, it seems like they are enriched in like bacteria which are there in gut. Okay, so it seems like they may be important. They are also there in many pathogenic bacteria and so on. Okay, now we solved the structure of first E. coli type 3 TA complex and which revealed its assembly mechanism. And based on this, we proposed a model of its assembly both in vitro and in vivo. Okay, so basically then with this I will conclude, and I come to my acknowledgement slide. I've already shown you the students who worked on this project, on these papers. Okay, for type 3 TA system HIC-BA complex, Pankaj and Vikrant. Pankaj has now his thesis submitted. Hopefully he will be defending it soon. And Vikrant is a postdoc in the lab. Chaitanya and Digvijay also worked in the lab during this project. Type 3 TA system, mainly the work I presented was done by Manikandan and helped by Kaveshri. And now Harshita, our new PhD student, has joined them to understand other aspects of this project. Okay, and a JRF in the lab, Sauradeep, also helped Manikandan during that time. And then I should thank my collaborator, Professor N. Srinivasan. We lost him last year, unfortunately, and he was actually, this TA project was brand new for us in IIC, right? So we did not come here to basically work on this. Our main interest is on telomere and telomerase biology, by the way. So this is completely different project, and with the enthusiasm of Professor Srinivasan, we got involved in this project. We became part of a big grant, a Center of Excellence grant from DBT, along with 6 other PIs, and that is how we started this project. Okay, and now we have some result to show to you all. And Sandhya in Professor Srinivasan's lab, characterized this. I mean, did this bioinformatic work initially that I showed to you on type 3 TA system. Professor Ramandeep Singh from THSTI Hyderabad is our collaborator. Okay, he helped us to get started on these bacterial growth assays for type 2 TA system, and his student Saurabh Chugh. And I have a friend, Dr. Uli Rothweiler, and a collaborator in University of Tromsø, Norway. And yeah, it is wonderful to have collaborated with him. And of course, I've showed you results from NMR, ITC, X-ray, and so on. And we have wonderful facilities in our campus for doing all this high-end work. Okay, so I would acknowledge these facilities. And of course, our funding is all from Government of India, from different grants that we have, and of course also from my host institute, that is IISc. Okay, so with that, I would like to end now. And yeah, if there is time for answering questions, I would be happy to answer. So thank you. Vinita?
Yes, so there are questions in the chat box.
Thank you so much, sir, for your talk. It was such an enlightening talk. So we have a few questions for you. So the first question is from Anjishnu Chakrabarti. And the question is, sir, is this toxin antitoxin system dependent on temperature?
Sorry. Just one second. Something is started here. Okay. So what is the question? Can you please repeat?
Yes, sir, of course, sir. So the question is, does this toxin antitoxin system depend upon temperature? Okay, I'm not able to understand what you exactly meant here, but if you mean that whether they are this complex assembly is dependent on temperature,
I don't know. And, they seem to be very robust complexes. Okay, they seem to be very robust complexes. So they can withstand some temperature. And, yeah, maybe what you are interested in, whether, some thermostable bacteria have these TA complexes and so on. We have not looked into this. Okay, yeah. So yeah, temperature dependent regulation of TA system I have not come across also.
Okay, thank you so much, sir. So second question is from Dr. Dhar. It seems this system activation is the last resort as it leads to the death of bacteria. Is that so?
Okay, this is from Dr. Dhar.
Yes, sir.
He's my good friend, and he was my notable. He's one of the notable junior from our college. So yeah, it's not a last resort. Actually, in fact, I, of course, in my talk I mentioned that bacteria can undergo altruistic cell death. Okay, when these systems are activated. But rather that seems to be rather rare. Okay, usually what happens is bacteria undergo into persistent cell state. Okay, so rather than they undergo this, in the restive state where they are just in the dormant state. Okay, so they don't actually die. That's a prevalent now understanding in this area that TA systems don't lead to actually killing of bacteria, but rather bacteria undergo in a dormant state, and they just wait there until the conditions are favorable, and then they come back. Yeah.
Thank you, sir. So second question from Dr. Dhar is, why would bacteria produce toxin, antitoxin and spend energy without any threat? Better would be to synthesize the toxin at the time of attack. Please elaborate.
Yeah, so I mean, this is again, we can only argue for one hypothesis over other, but in my opinion, keeping something, if it is very important for bacteria to survive under certain conditions, it's better to be ready. So at that time, bacteria basically keeps all these system in them, and maybe it is faster to activate them from the complex. Okay, when you read the review on this line of research, you do find that there is a fitness cost, okay, for bacteria. And so in one of the slide that I showed in the very beginning, different mechanisms of antiviral strategies in bacteria, right? I mean, a single bacteria will not have all those mechanisms, right? So bacteria, they will have only a few antiviral strategies in them. Okay, for example, actually the TA system in our lab strain of K12 strain, right? We have not been able to identify this TA system, type 3 TA system. So yeah, bacteria don't keep all the system ready, but they do keep enough that they survive. Okay, and yeah, that's how I can answer this.
Thank you, sir. So the next question is from Amrita Parna Methi. What do toxin and antitoxin system have effect? Sorry, sir. What effect do toxin antitoxin systems have on bacterial genome?
I mean, so I showed you in the beginning that one of the way people thought that they function or their role is to maintain the plasmid. Okay, so many bacteria have, of course, its own genome, and also some of the genes are found in plasmid, right? On plasmid in bacteria. So how bacteria maintains those plasmid, also had been an area of research, and people found that by keeping TA system there on the plasmid, bacteria make sure that these plasmids are propagated. Okay, so that is the thing I can. But later, people did find several of these systems on the chromosome. Seems that they are integrated into chromosomes, right? So so yeah, that is how I can explain that they do have role in maintaining certain gene from getting lost. Yeah.
Sir, so the next question is from Siddhant Sharma. Piggybacking off the above question, can there be TAS gene that may be phase in the origin but reside in bacterial genomes?
Okay, this whole thing is actually quite upcoming. Okay, there are some theories now which are pointing to the phase origin of these genes. Okay, actually you are right. So maybe you go back and you can look at these provoking ideas. Okay, So yeah, I have come across some reviews where they are saying that they may have, at least some systems may have actually phage origin. Okay, but again, even my knowledge is limited on this, so I'm not going to elaborate further or speculate rather further. But yeah, it seems like yes, they may have a phage origin also in some cases.
Thank you, sir. So we have another question from Dr. Dhar. Do all bacteria have TA system? If yes, can these be used as an effective antibacterial? This can therefore take care of AMR.
Yeah, exactly. So yeah, so as I mentioned, there are now 8 types of TA system classified. Okay, so there are a lot of type of TA system. Okay, there are 2 component system, right? Of different mechanism and so on. So I can fairly say with the confidence that all the bacteria will have TA system. Not all the type of TA system, but some form of TA system every bacteria has. Okay. Now, of course, for example, type 1 versus type 2 TA systems, you can't have the same molecule now inhibiting both the systems. So, but yeah, you can. That is the motivation, that if we can study some system in pathogenic bacteria and find a inhibitor of this complex, right? Complex assembly, or rather we call it activator of these assembly in bacteria, we can that can be an effective antibacterial strategy. And that's why people call them like ticking time bomb in bacteria, because you can actually unleash this time bomb against bacteria itself, right? I mean, under stress it happens in bacteria, but can you do it with a a molecule, small molecule or a peptide based inhibitor? That's I think many people are thinking about that and doing this, right? So definitely it can be a antibacterial strategy.
Sir, the next question is from Tanya Malhotra. How can including suicide molecules against pathogens could help in combating the growing problem of multi-drug resistance?
Yes, this is the same thing, same question which the previous question. Yeah, we have to look for new ways, ways to kill bacteria or inhibit bacterial growth, right? Because we are running out of antibiotic. We all know that it's a real threat. I mean, you will, I have come across, for example, my mother was, found to be resistant to most of the bacteria. It becomes a scary situation suddenly, right? In a hospital, whenever there is hospitalization or infection, right? So yeah, this antibiotic resistance is a real threat. Okay, and we have to find new ways, novel ways to, novel antibiotic, right? And TA system definitely is one system. Now, of course, several people are working on this idea to activate TA systems and see whether they then, you can inhibit bacterial growth, right? That will be quite novel way. But, I must say that, because there are so many type of TA systems and they also crosstalk to each other, it has been rather challenging. So if we, you will have to find, they basically, they something else will take over its function and so on. So in our larger grant, for the grant that I mentioned, right, I was part of it. That was that is what they were trying to understand, this crosstalk between different TA systems and so on. But yeah, in principle, it is possible that we use these systems to find a new molecule for as an antibiotic.
Thank you, sir. Sir, so the next question is from Monica Bhardwaj. So what things must be kept in mind for selection of model microorganism to study toxin antitoxin systems?
So, okay, if I understood the question properly, so yeah, when we propose this grant, if you want to understand now something in E. coli, I mean, usually, people will find what is the point. I mean, you want to study something in pathogenic bacteria like MTB or other pathogens, right? So yeah, that is the motivation. You want to study them from the pathogenic bacteria. In type 3 TA system, when we started working, we did our searches and we could not find any type 3 TA system in MTB. Okay, so that was our model organism. We could not find it there. We still cannot find any type 3 TA system in MTB genome. So then we looked in E. coli, and we also know that, for type 3 TA system, not many things were known about its assembly, structure, and so on. And working in E. coli has its own advantage, right? At least you can establish basic pathways and so on in E. coli. So we resorted to E. coli, and but yeah, from moving on, even for us, we would like to look at these the system from pathogenic bacteria. That is what I would do, in order to make a more impactful work.
Thank you, sir. So this question is from Dr. Amrish Saxena. Is this target druggable? Are there any known peptide-based therapies either in clinical phases or in commercial phase? If not, what would be the challenges in designing the inhibitors? Yeah, so Amrish is also my my dear friend and my junior. So thanks, Amrish, for being here. So yeah, it is. Several people have tried actually, and in principle, people have tested peptide-based inhibitor to disrupt the TA complexes, and in principle, it works. Okay, there are papers where people have found and so on. Now, to develop them into a drug is another challenge altogether, right? So I'm not actually aware whether TA-based drug is there in any clinical trial or any phase 1, phase 2 trial. I'm not aware of it, but it has been challenging. Okay, so especially protein, the type 2 TA system, which are rather better studied, right, where it is a protein-protein complex, mimicking, disrupting protein-protein complexes which involve larger surface, right, is actually challenging. You have to, disrupt a large surface, right? So there are challenges, but in principle, people have actually, there are papers where people have proposed that they can successfully disrupt and so on. Okay, that's why type 3 TA system we think have more advantage, because this is RNA protein interaction which are more specific, and maybe you can mimic that RNA with another antisense RNA and so on. We have actually some ideas which we ourselves are also looking at right now. But yeah, that will be my answer. People have tried. In principle, it works, but I don't think something is there in clinical trial yet. Thank you, sir. So the next question is from Vinita ma'am, Vinita Kashyap ma'am. Has anyone used CRISPR-CS system against TA system? I don't know, but I am actually not. I don't understand the question also fully. So I would say
I wanted to ask about the CRISPR-Cas9. Because it's a RNA, you are talking about type 3, which is having the antitoxin as RNA. Correct. So that's why I was just intrigued that can that be used?
Yeah. So I mean, see, CRISPR-Cas or the restriction modification system, they have been thoroughly used for other biotechnological applications and so on. And so this TA system also holds some potential. We just don't know for what purpose we can use them, right? So, of course, one is that we somehow can make it druggable, right, and use it. So yeah, but definitely, in the type 3 TA system, at least, there is also RNA playing important role for the assembly of this complex. Okay, so yeah, it is also an ancient sort of system in bacteria, it seems, just like CRISPR-Cas system for fighting against some viruses.
What is the size of the whole complex? Because it was forming a hexameric complex.
Yes. So the whole complex size, it will be here. Okay.
Yeah, this one.
Yeah, I think this one. So, right, yeah. So this is it is written here. It's about 100 kilodalton. Yeah, 3 RNA and 3 protein in this case. Yeah. So this will be about 100 kilodalton. See, the RNA is just one repeat, which is about 33 nucleotide, and protein is, I think, 15 or 16 kilodalton. And so of course, they form this heterohexameric assembly. But this is one assembly, by the way. We believe that there will be other type of assembly possible. If something is forming a cyclic complex like this, right? You can imagine that you can form a now starting with a heterotetramer, right? You can form even hexamer, pentamer, and so on, because it's a cyclic, right? You just have to add one more molecule. I mean, from trimer to tetramer, tetramer to hexamer to octamer, and so on. So these are also some questions which we feel ourselves interested in, is what determines the size of this complex, right? So why not to add one more RNA and one more protein to make it bigger complex? And what is the meaning of this this cyclic complex? So yeah, we have basically unanswered question right now for this, but yeah, we wonder about this.
It's a very interesting work you have been doing, and I think more and more you go deep into it, you will get the answers of new findings. Actually, you will be the first one to solve many of the queries.
No, I mean, I must add that for type 2 TS system, there are several structural work already done by other group. On type 3 also, we are not really the first one. We solved from the E. coli. The people had actually studied from other bacteria like Pectobacterium and so on, because in common bacteria like MTV and E. coli, it was not there when we started the work. So then people found it in all these unique bacteria and studied them there. But definitely studying E. coli system would now, I hope, will make many experiments possible to do. Yeah.
Thank you, sir. That was all of the questions. I thank you so much, sir. No, sir, I don't think there's any other question. Yes, sir, that was the last question.
Am I audible?
Yes, sir.
Yeah.
Thank you so much, sir, for your enlightening words. We learned several new things about the significance and application of toxin antitoxin systems. I sincerely hope that many of us would be inspired by your work to take up further research in this field. Sir, we regret that we were unable to facilitate and hear you in person due to the current COVID situation. We would love the honor to listen to you in person. However, in the light of current situation, we would like to present you with an e-memento and e-certificate, sir.
Oh, this is super nice. Oh, thank you so much. Yeah, so sure. I mean, it will be my honor to visit my college back and meet all the teachers. So lovely to see, so nice to see all my teachers here, especially Meera ma'am, Rajni ma'am, Kamna ma'am here. So by the way, ma'am, yeah, I mean, among my colleagues, I married then Tina. So she's
okay. All right.
Oh, very good.
Yeah.
And, what is she doing?
She's doing
Tina
good.
kya kar rahi hai?
Yeah, she's now she's at INSTEM NCBS campus. She's a faculty there.
Oh, so, very good.
Yeah.
That's
congratulations. Good. Very good.
Yeah.
And our other colleagues are doing pretty well. Of course, Sachin is now entrepreneur. He's doing very well. Richa is there in US, also now a faculty, I think. I think I'm aware of only some of them, yeah, but I'm sure they all are doing very well.
Richa kaha hai? Where is Richa?
I think Richa is in New York right now.
Yeah. Good, very good.
Yeah. We came back in 2014, so it's been some time now.
Making Bharat Atmanirbhar Bharat. You are back to India, yeah, very good.
It was very nice listening to you, Mahavir. Very nice.
Thank you so much, ma'am.
I'm so happy to see you do so well. All I can say is that as students, when you ask questions, you teach teachers. I'm not asking you any question because I'm not going to be able to teach you anything now.
No, ma'am. I mean, yeah, but it was wonderful. We had wonderful time there.
Yeah.
I
remember your batch.
I hope you remember our batch,
I do.
Yeah, Sachin was our star boy.
Sachin and Mahavir, I remember you also. Though if I'd seen you outside, maybe I wouldn't have recognized you. But now it's good.
Yes. We have changed.
And I also like the way you mentioned about the Biospark thing. I remember It was,
Yes.
the name was given, Biospark and all that. How
So-
much thinking we did at that time. Yeah.
Yes, yes,
It's
yes.
good.
So, that is how I got in touch, I think, with Vinita. And so, because I saw on Facebook that they are doing Biospark, and it caught
Okay.
my attention that, this is what we had done.
Yeah, we
first time.
actually started the name Biospark at that time.
Biospark also that year, yeah. And I think, Professor Anil Tyagi and Syed Hasnan,
Yeah.
we had
They-
invited them.
Yes. yes.
And, we had quiz and, model making and all that. And
I know.
even,
And it was
got first
good.
prize.
It was inter-college thing and it was very
Yes.
good. I think I was quite proud of it at that time.
Yes,
We-
yes.
We went to the, it was also held in Daulatram College also at that time.
Maybe
Biospark.
they gave some other name, maybe.
Yeah, maybe in my time now it was in Daulatram.
Okay.
Inter-college competition.
Yeah.
No,
yeah.
competitions तो शुरू से ही होते थे from 1988, but to this
Yes,
level
yes.
it was different.
Yeah.
And
Yeah.
the name is given to a festival and all that. Otherwise, you could have paper reading contests, some debates like that
Yeah. Yeah.
in inter-college.
No, we all, I mean, Sachin was very active. So, we went to.
I know
different
Sachin was
companies,
very
got some,
active.
sponsors and, all that. So it was a lot of activities. It was fun. And ma'am, yeah, I also remember you taking us, for reading using spectrophotometer to identify. We would actually
Yes.
take all of us to go there all the way. That was the effort.
To
I mean,
IIT.
Yeah.
Yeah, I remember because that I had to take readings. I had to ask my husband, please let me
Arrange
use your
something.
spectrophoto. It
Yes,
was so
yes,
funny.
Yes.
I remember doing that.
Yeah.
Yes.
So
yeah.
I mean, as I think one of teacher mentioned that now we have better facilities.
I know. Now they are doing very well.
That's great.
Those were the days.
But then they don't get to go to IITs, students. So we used to be so happy, to go there and.
Yeah, I know. You could see
With
the
you.
whole thing. Come home when you are in Delhi.
Sure, ma'am. Sure. Yeah. When I'm in Delhi, I mean, because of this
Come over.
corona and all that,
Yeah. Come home.
we're not able to travel. I thought I will flash some pictures and so on, but they're all in Delhi, also in my home in Delhi. And
Okay.
now, now we don't have them in digital form, no? I mean, we have to scan some albums. So I could not flash those. Yeah.
Very nice.
But
Thank
it will be wonderful to visit all of you. Yes.
Thank you.
Yeah. Thank you, ma'am.
Thank you, sir. I would also like to request you to join us at the Sci Idea event. We would be starting it with Sci Idea event now.
Okay.
Greatest scientists are artists as well. This is a quote from Dr. Albert Einstein. And with this, I would like to welcome you all to the Sci Idea, the inter-college competition for designing cover page of Biospark 2022 magazine. The winner of this competition will feature as the cover page of Biospark 2022 magazine. Science and technology capabilities are fundamental for social and economic progress in developing countries. Modern India has had a strong focus on science and technology, realizing that it is a key element for economic and societal growth. With the advent of Atmanirbhar Bharat campaign in 2020, it has become essential for us as a society to focus on the contribution of science towards this goal. Keeping this in mind, the topic for this year's competition is the role of science and technology for an Atmanirbhar Bharat. For this competition, we have received 19 entries from the students of different colleges. In this competition, participants had to make an A4 size colored or black and white cover page. The cover page would feature brief details about their idea. However, the judgment would be primarily based on the innovation and design. I would now be presenting these cover pages that the students have submitted. And for this competition, we have our judges, Dr. Manisha ma'am, who is the teacher in charge of Zoology Department at Deshbandhu College, Dr. Aparna ma'am, who is the teacher in charge of Botany Department at Deshbandhu College, and also our senior professors of Biochemistry Department, Dr. Kamna ma'am and Dr. Rajni ma'am. I hope my screen is visible and everyone would be able to see the posters.
Yes.
Yes, ma'am. So this first poster, this first cover page, I'm so sorry. This first cover page has been submitted by Muskan Agrawal, who who is a third-year biochemistry student at Deshbandhu College. This cover page, this cover page has been submitted by Prem Sai Biswal, who is a first-year student of biochemistry at Deshbandhu College. This is his idea. This cover page has been submitted by Pooja Kumari, who is a second-year student of biochemistry at Deshbandhu College. This is her idea. This cover page, I'm so sorry, I'm saying poster again and again. This cover page is submitted by Bhumika, who is a student of biochemistry at Shivaji College. This is the idea that she has submitted. Moving on to the next cover page. This cover page has been submitted by Pankhuri Gupta, who is a third-year biochemistry student at Deshbandhu College. This is her idea. My screen is visible to everyone, I hope. There is no problems. Moving on. This poster has been submitted by Tanya Malhotra, who is a second-year student, and this is a cover page idea. She's a second-year student at Biochemistry Department, Deshbandhu College. Moving on. This cover page has been submitted by Soumya, who is a zoology student at Deshbandhu College. Moving on to the next cover page. This has been submitted by Tanya Jan, who is a second-year biochemistry student at Deshbandhu College. This is her idea. Moving on, this cover page has been submitted by Divyansh Yadav, who is a second-year student of Biochemistry Department at Deshbandhu College. Moving on, this is a cover page which has been submitted by Afaf Salim, who is a third-year biochemistry student at Deshbandhu College. This cover page has been submitted by Priyadarshini Pari, who is a first-year student of biochemistry at Deshbandhu College. This cover page has been submitted by Siddhant Sharma, who is a third-year student of biochemistry at Deshbandhu College. Moving on to the next cover page.
Cover page designed by Bhavya Sutha, who is a third-year biochemistry student at Deshbandhu College. This cover page is designed by Asha Lakra, who is also a third-year student at Department of Biochemistry, Deshbandhu College. This cover page is designed by Monica Bhardwaj, who is a first-year Department of Biochemistry student at Deshbandhu College. This cover page is designed by Noni Sharma, who is a third-year biochemistry student at Deshbandhu College. Lastly, this cover page is submitted by Cynthia Mondal, who is a first-year biochemistry student at Deshbandhu College. Thank you.
Thank you. So these were the posters which were submitted for the cover page designing competition. What is an amazing display of creativity by all of the participants. With this, we reach the end of this competition. I would now like to invite Dr. Manisha ma'am and Dr. Aparna ma'am to say a few words and announce the results of this competition.
Okay, good evening, everyone. As per the posters, sorry, the cover pages, the selection was very hard, and the students have really displayed their best, and they did a wonderful job. And their thought process and their selection of the material for the cover page was really worth commendable. It was really commendable, and I'm really impressed by the talent of the students. And over to you, Siddhant, and that's all I wanted to say. Thank you so much for making me a part of this very important decision. Congratulations to all the participants, especially all the participants, and also to the winners.
For your words. So without any further ado,
Aparna ma'am is there. She was there.
he is not able to unmute.
Ma'am, are you able to unmute yourself now? Kamna ma'am?
Yeah, I'm already unmuted.
Ma'am, Aparna ma'am was here, but I don't know.
Aparna has joined.
Yes, ma'am, she has joined, very much joined. She was there, ma'am.
To invite her.
Ma'am,
She has joined. She has joined.
Aparna ma'am, are you able to mute yourself? Unmute yourself. Ma'am, she is writing that there is some network issue and she is not able to unmute herself.
Has she given the result?
Yes, ma'am. Then, ma'am, Manisha, ma'am.
Yes, Vinita.
Ma'am, I've just, I mean, you have the results, ma'am. You can you announce the result, ma'am?
Announce the results, Manisha. You please
Yes.
announce the results.
Okay. I'll just have a look. Just a minute. Yes, I'll just see them. It's gone.
Yes, ma'am.
Okay. Yeah, I have seen the results. Am I audible?
Yes, Manisha, yes.
Yeah, okay, ma'am. So the results go here. Muskan Agarwal takes the first position. The second position goes to Asha Lakra. And the third position goes to Bhumika from Shivaji College, and the above two students are from Deshbandhu College itself. Congratulations, students. Really very nice it was. Many, many congratulations.
Kanish, can you present their sketch again for all the three
Yes,
winners?
yes,
For all the three winners once again. Muskan's first, who is the first winner?
yes, ma'am, I'm presenting. Ma'am, I hope it's visible now.
Yeah, it's visible.
Yes, it is.
Muskan is already, I think, in Art and Craft Society. So being from that society, she has like put all her best efforts in designing the cover page of the Biospark magazine 22. So it has come out really well, really extremely well among all the participants. So that is why first prize for, from all the 4 judges has gone to Muskan Agarwal, third year student.
Are you there?
Congratulations, Muskan.
Big congratulations to Muskan Agarwal. Muskan, are you there?
Yes, ma'am. Thank you so much.
You want to say something?
Yeah, I just want to quickly explain what I have
Yeah,
done.
sure.
So
Sure.
the topic was Atmanirbhar Bharat, and I just tried to show how India is working towards an Atmanirbhar Bharat in various fields, from agriculture to space and research to medicines and now to education as well. We are really trying our best to make our resources by ourselves and lead India to an Atmanirbhar Bharat. So this is what basically I tried to show in my cover page.
So congratulations once again, Muskan. It has really come out extremely well. Extremely well. Yeah. All 4 judges like this one. Now the second one. Kanish, can you present the second cover page?
Yes, ma'am. Ma'am, the second prize winner was. Yes, ma'am. Yes, ma'am. I'm reaching.
Then hers is in the last.
Yes, ma'am.
Last but one.
Yes, ma'am.
Yeah, this one.
The second prize winner, ma'am.
Ayush, can you unmute her? Asha, can you unmute yourself?
Yes, ma'am.
Asha, explain me. What was the idea when you were doing this sketching?
Actually, ma'am, I searched for the inventions that are invented in India. So to show the inventions of India, which makes India to be Atmanirbhar.
So what areas you have covered here in this picture, in this cover page?
Ma'am, technology, agriculture field, vaccines, and some inventions that help our India to.
Why didn't you use colors while drawing this?
In my home, there is no color. That's why I make black and white.
Then it is acceptable. Nice. So congratulations, Asha, for winning the second prize. Now the third, Kanish, show the third one.
Yes, ma'am. Ma'am, I'm showing.
Bhumika is not here at the moment, so she'll not be able to interact with us.
एक बार show कर दो.
Yeah, yes. We'll show the.
Ma'am, is my screen visible, ma'am?
Yeah, it's visible.
And this one. Yeah.
Aparna now, and Aparna, she could unmute herself or not? Aparna.
Yes, ma'am. Am I audible?
Aparna, and then say something after this is projected.
Yes, ma'am. Yes, ma'am.
Share your words. So, this candidate is not around, so you can only see this picture. And then you can invite Kanishk, you can invite Aparna ma'am to say a few words.
Yes, ma'am.
Then Rajni ma'am also as well, finally.
Yes, ma'am. Ma'am, I've stopped my screen share.
Yeah, you can stop.
Congratulations to all of the winners. Now I would like to call up Aparna ma'am to share a few words with us.
Am I audible?
Yes, ma'am.
Yeah, Aparna.
Yeah, good evening to all of you. First of all, I want to congratulate the whole biochemistry team, Dr. Kamna ma'am and Dr. Rajni ma'am, and the conveners, Dr. Vinita and other organizing team, including the students, for organizing such a nice lecture from your alumni, Dr. Mahavir Singh. And it was really wonderful to hear him and to hear our own alumni from the college who is making us proud. And as Manisha ma'am said, it was a very tough competition to judge all the participants, and they did means wonderful. They have presented very well, and a lot of creativity they have shown while creating that poster. And even the themes were also very well defined. So it was a bit difficult for me to judge all those cover pages. But so I wish them all the best. Congratulations to all the winners as well as the participants. All of you have done wonderful job. And though because everyone can't be the winner, but all of you are, to me, I think all of you are winners. So congratulations and all the best, and best wishes to the Biospark team also. Thank you, ma'am.
Thank you so much, ma'am, for your kind words. I would now like Dr. Rajni ma'am to share a few words with us.
Anish, if Meera ma'am has not still left, invite her as well. And
Okay.
Rajni ma'am also. If Meera
Yes.
ma'am has not yet left, then find it out and then call her.
Yes, ma'am.
She's here.
Mahavir sir has left or still there?
Yeah, sir is also there.
अच्छा हाँ तो सर को भी एक बार सर को भी मतलब can say something regarding all the
Absolutely.
three
Yes,
these.
ma'am. Yes, ma'am.
Shall I say?
हाँ, sure.
Yes, ma'am.
Unmute ho na? हाँ. Congratulations all the winners. The ideas was very innovative and the drawing was excellent. I cannot really imagine that our scientists, our future scientists are, they are so, such a good artist they are. Such a hidden art is there in them. I mean, they have everybody has done wonderful job. Their ideas were very innovative, creative, and the sketches were very well drawn, very beautifully colored. Muskan has done a wonderful job. And I like all the entries. All the entries were very good, and they were depicting the Atmanirbhar Bharat. So the idea was very good, and the theme was very good. I would really congratulate all the students who have won the prizes, as well all the entries which we received as entries. So I congratulate everyone, and I wish you good luck for your future endeavors. Thank you so much.
Thank you, ma'am, for your kind words. I would now like to call Dr. Mahavir Singh to share his thoughts with us.
Yeah. Hello, everyone. So yeah, it is just wonderful. I've seen all the artwork. I'm just blown away. I'm really nice pieces of drawings that you guys have, especially the thought process behind. So yeah, all the my best wishes to all of you, and congratulations to winner. All the artworks were actually wonderful. I think judges had hard time to pick up those three, but they all were wonderful. Okay, and yeah, let me also wish you for for Biospark 2022. I hope you will have a great Biospark. So I think this is meant for this activity will happen for the entire year, right? So yeah, and all the best for your coursework. Do well, and yeah, it was wonderful to be with you guys today. Thank you.
Thank you, sir. I would like to invite Dr. Meera Kaushal ma'am to share her thoughts with us.
Dr. Meera is there? Is she there?
She is there.
Hello, Mahabir. Mahabir. Hello, Dr. Mahabir. हाँ, जब आपने talk end किया तो I was not able to unmute myself. That is why I could not say anything. But your talk was really fully relishable by all of us.
Thank you, ma'am.
All the students, I think they enjoyed your talk. So thank you for being with us today, and thank you for sparing your such a precious time with all of us.
My pleasure, ma'am. Yes.
And near future also we hope.
महावीर, मेरा एक क्वेश्चन है, मैंने पूछा नहीं उस समय.
Yes,
ये एंटीटॉक्सिन और वैक्सीन, they are same?
Antitoxin and
vaccines?
No, ma'am. No. These are no.
It cannot be.
No. These are just, these are bacterial genetic module, right? So they seems to play important role in their in several aspects of bacterial physiology. So yeah.
Okay. Vinita, फिर अब आपका क्या final है? Meera ma'am is not, I think, is not there. So let's end it. और आपका क्या end में रहता है? So do that part and finish it. Vinita, can you hear me?
Yes, ma'am. Sorry, I forgot to unmute myself. Ma'am, Tanishri gave a vote of thanks. Thank you very much, Dr. Mahavir. You were very kind and considerate. He is a very gentleman, and on the very first go, I mean, I didn't have a known. I just came to know him from Dr. Devjyoti's talk only. I didn't know anything about him, but on my first.
Meera ma'am का message आया है. Vinita, Meera ma'am is still there. She's writing thanks, but she's not able to join. What is the problem?
And then I have to call her actually. Should I call her, ma'am?
I don't know whether she's there because Kanishk also called her no, that time.
Yes.
She was there till now. Till the
She's
end,
there.
she was there.
Writing.
I was seeing her.
Writing.
I was there. She was there.
Or you, all of you were supposed to take the group pic also.
Can you join?
Yes, ma'am. It's.
Can you join?
Then invite her if she's there.
मैम वो नहीं है.
अच्छा ठीक, ग्रुप पिक ले लो फिर एंड करो.
She is coming, ma'am. I just called her. She is rejoining, ma'am. She left by chance. So
Okay, she's always welcome.
So I was just telling ma'am that on the very first go, sir was like so receptive, and he didn't. I mean, I even did a mistake. I didn't tell to anyone that I had actually, instead of Friday was the commitment, and by mistake I have written on the poster 29. And in spite of that, he was so, I mean, so much cooperative that he was still agreeable, and he didn't refuse at all. So I mean, really, I'm very thankful. And sir, from the bottom of my heart, and I would really like to continue the, in fact, I have told my students to, go and, join your lab, because I don't know, I really got inspired after, because your work is new, and many things can be still done in that. So in fact, I'm like pushing all the students to go and join your lab. These are the ones who are there in India, who don't want to go out of India.
Yeah, thank you.
Thank you, Dr. Mahabir, for being with us once again.
Yeah.
So take a group pic then, Vinita.
I'm just for 2 minutes because Meera ma'am would be rejoining, she said.
Okay. Preeti did not join, huh? Preeti?
Ma'am, it will be very early at her place. Almost 4:35. So I don't know, ma'am.
Ma'am ne join kar liya. Dr. Meera has joined. Invite her once.
Yes, ma'am. I would now like to invite Dr. Meera Kaushal, who is our retired faculty of Biochemistry department. Ma'am, please share your thoughts with us.
Ma'am, are you able to unmute yourself? Meera ma'am? Yeah, she is there, ma'am. Meera ma'am, can you listen?
I'm trying to, beta. I'm trying to.
Okay.
Video nahi aa raha. Anyway, well, thank you so much for inviting me. I'm so happy to see Biospark doing so well. And like Sushant, his name, no, what was his name? Sharma, kya naam? Siddharth. Siddharth Sharma. Like he said that it's the students are learning a lot and there's a lot of creativity. They are bringing it out. That's what actually, when we started Biospark, we wanted that. So that's what is happening now. And I'm very happy. And I'm very, very grateful to you all for inviting me. Every time you invite me, and I love to come. I wasn't feeling too well today. I'm back in my bed, actually. If you see the video, I don't know. It's not coming. I'm back in my bed. Anyway, it's not coming, which is odd. Oh, it's come. Anyway.
It's coming, ma'am.
It's come right now. I'm there. I'm not feeling too good, but I love to come and listen to you all. And it was very well organized. It's, look at Mahavir. He's doing so well. I'm really proud of him. And of all of you too, the whole Biospark team and all the teachers and all my colleagues. And of course, Vinita is my student too.
Ma'am, Biospark is your creation as well as your child as well, your child.
Yeah, it is my child and I love it
your
too.
creation. That we are continuing.
I know, Rani, but it's all because of you that you are doing it. You see, you are the one who is bringing it up.
Grow kar rahe hain aapki child ko.
That's what you are doing. And you're doing a good job. I'm proud of that. That's what it is. I'm proud
Thank
of
you, ma'am, for being with us.
I am so glad. Thank you, Vikram.
You were unwell, but still you joined.
No, I'm not. My throat is bad and so, but it's not COVID. That's the good point. Yeah, I don't have fever. Yeah.
Good.
Thank you so much, बच्चों, and keep it up to the students. Biochemistry. आप नहीं जानते हो, मैं बहुत tough भी थी जब मैं थी. Good, I'm not there now for you. Anyway, do well, बच्चों.
मैम ऑनलाइन में आप tough नहीं हो सकते थे.
उतनी टफ नहीं थी मैं. But anyway, I want अच्छा खूब मेहनत करो and खूब करो. As I always say, they've all heard me, sky is the limit. You have to go very high. Go, keep on, go and fly. Thank you for inviting me. Thank you.
Please stay here for a group photo, ma'am. Please don't discuss.
Arey wah. Sure.
Just please wait.
Sure.
Yeah, Kanish, please thank you.
Yes, ma'am. Thank you so much, ma'am, for your kind words. Once again, good afternoon, everyone. It is such an honor for me to get the opportunity to thank all of the dignitaries. I would like to express my gratitude to all esteemed delegates of the webinar for their presence and contribution to make this event a great success. I would like to thank our respected Principal Sir, Professor Rajiv Agrawal, respected Vice Principal Sir, Professor Kamal Gupta Sir, respected Teacher in Charge Ma'am, Dr. Kamna Singh Ma'am, Honorable Coordinator IQAC, Dr. Aditya Saxena Sir, Honorable Coordinator DBT Star College Scheme, Dr. Indrakant Singh Sir, respected judges, Dr. Manisha Ma'am and Dr. Aparna Ma'am, our senior faculty, Dr. Kamna Ma'am and Dr. Rajni Ma'am, who were also the judges for the program, and a very special thanks to our retired faculty member of Biochemistry Department, Dr. Meera Ma'am, for joining us today. On behalf of Department of Biochemistry at Deshbandhu College and the entire Biospark team, I, Kanishk Singh, would like to express my heartfelt thank you to our esteemed speaker and alumnus, Dr. Mahavir Singh, for sharing his discoveries and perspectives with us today. On the topic Structure and Assembly of Toxin Antitoxin System, Intracellular Molecular Time Bombs in Bacteria. It was a very interesting topic, sir, and we are very grateful for you to give a lecture on it. In today's webinar, we discussed about the significance of the toxin-antitoxin systems, how toxins are neutralized by antitoxins, why bacteria carry antitoxin-toxin systems, biochemical applications of toxin-antitoxin systems, and the role of antitoxin-toxin systems in antiviral mechanism, and much more. Thank you so much, sir. I would like to express my gratitude to Dr. Kamna Singh ma'am and Dr. Vinita ma'am, the convener of Biospark events, for organizing another fascinating webinar. I would also like to thank the faculty members, Dr. Rajni Jain ma'am, Dr. Preeti Karwal ma'am, Dr. Vinita ma'am, Dr. Prabha Arya ma'am, Dr. Vanshikalam ma'am, Dr. Astha Giri ma'am, Dr. Garima Singh ma'am, who are among the faculty members who I'm very grateful. And also a very special thanks to Dr. Meera ma'am also for starting this entire Biospark event in the first place. Also a very big congratulations to the winners of Sci Idea competition Finally, and most importantly, thank you everyone for participating in this webinar and making it a great success. I sincerely hope you liked it. Now I'd like to ask everyone to turn on their camera so we may snap a group photo.
There is a feedback link also in the chat box. So I'll request all the students to please kindly fill it before they leave. Or they can, after the group photograph, we'll stay there for 5 minutes. They can still fill the feedback.
Ma'am, your mic is muted.
विनीता हम लोग को भी विनीता हमने भी भरना है फीडबैक फॉर्म.
Yes, ma'am. If you, you can. I mean, you should.
अच्छा नहीं तो सर्टिफिकेट नहीं मिलेगा.
नहीं ma'am, not that way. That way, ma'am, but that app is like that, ma'am, that if you fill it.
मैम पार्टिसिपेशन का नहीं मिलेगा फिर बाकी मिल जाएंगे आपको.
फिल कर देती हूँ. नहीं ये भी तो बैठी है पार्टिसिपेशन में तुम्हारी छुटकी सी. इसको भी मिलेगा पार्टिसिपेशन का.
इसको पिक्चर मिलेगी. इसको पिक्चर मिलेगी मम्मा के साथ.
Group photograph. Can you take the group photograph?
Yes, ma'am. I'll be taking the group photograph. So I'll request everyone to turn on their cameras, and I'll say 1, 2, 3, cheese. At cheese, please smile. So 1, 2, 3, cheese. Yes, ma'am. I've taken the group photograph.
Thank you. Thank you.
Thank you, everyone.
Thanks, Mahaveer, for such a good lecture.
Thank you, ma'am.
Thank you, Mahaveer. It was such good. Now, when you come to Delhi, come home.
Sure, ma'am. I'll get in touch with you. Yes.
Yeah, please.
Take
So
care.
we all are in Delhi, so you have to visit.
Yes.
We'll have a grand party.
I know we could. Let's meet in college also. We can go back. Yeah.
Yes, ma'am. Yes. Okay. Thank you, ma'am. Then I will leave now, all of you. Thank you again. Okay. Yeah.
Thank you. Thank you so much.
Thank you so much, sir.
Okay. Bye now.
I think it's over now, right? Rajni, अब जाएं?
हाँ नहीं उसमें ना feedback form भर देते. We can fill the feedback form.
Where is the feedback form?
I'm in the chat box.
Okay.
Chat box में link दिया हुआ है.
I'll find it and
They have sent a link.
Okay. We'll see. I'll see. ठीक है.
I'll post it again for you because you left so many.
Okay, beta. See you. Thank you.
Bye, Meera.
Bye, Rani.